All right, thank you very much. Hope everyone can hear me okay. And apologies, this weird method of presenting, but I hope that everyone enjoys. It's such a wonderful thing that we can still do it. We have the technology and we can share our ideas and data. Speaking of data, Dennis has told me that he's looking forward to seeing my data. Um, I'm not going to show data. So he asked me to talk about reproducibility, replicability, and so I'm going to focus on this question. It's kind of a general, more general talk, less data. I wanted to make it relatively easy and not a lot of details, but I think it is a very important topic. So hopefully you guys will enjoy it. Okay, so um, let me go to the next slide. Let me start with the definition. These colloquial terms, they seem very similar, but they have actually different meaning. So reproducibility and replicability are terms that we often use, and we all think about, they mean basically that we need to replicate things. We need to find something similar if we repeat the studies. The actual definition of reproducibility is the strict one. This is what it means is that reproducibility is obtaining the same results repeatedly when using identical methods. Replicability has kind of a looser meaning. It means reaching similar conclusions even when variations in methodology exist. So I want you to remember this. It's not just semantics, it's not just splitting hairs and talking about these words, what they mean, but they actually will come back to us at the very end in our conclusions. So again, remember, reproducibility is about obtaining the exact same results using exactly identical methods. Replicability is about getting roughly the same conclusions when we are using, even when we are using different methods. So arriving at the same or similar conclusions from different perspectives or using different methods. All right, why are we talking about this? What's the problem? Well, a lot of people talk about reproducibility crisis. So if you look at the popular media or reputable even top tier scientific peer-reviewed scientific journals like Science and Nature, you find these headlines. So for example, the New York Times headline says, many psychology findings not as strong as claimed, study says. Nature, over half of psychology studies fail reproducibility tests. Another newspaper, scientists replicated 100 recent psychology experiments, more than half of them failed. Science says only 36% of studies replicated. Nature, massive international project raises questions about the validity of psychology research. And then there is this questionnaire filled out. Nature initiated this, and they asked scientists and students, and 52% of the people who they asked say there is a significant crisis, and 38%, additional 38% says that there is a slight crisis, and only 3% said that there is no crisis. So clearly something is happening in science that people don't like. There is a problem with how we replicate things. So is science trustworthy? Some people ask. Now here's another example of how serious scientists and everyone takes this issue. Entire conferences have been held designed to address the crisis. There is one, replicability and reproducibility of discoveries in animal phenotyping. This conference was focusing on how we measure animals. Mainly behavior geneticists participated. It was held in Israel, in Tel Aviv University about 3-4 years ago. And it covered a huge range of topics, how we can improve replicability, reproducibility, what are the problems, what we can do to make things better. Um, Here's another warning sign that things are not going so well. Even the NIH issued guidelines as to how to achieve reproducibility. So this is the actual website, the web page, copied from the NIH's web page into this presentation. And they are worried about how to enhance reproducibility and how to find like they provide guidance how to make our experiments more rigorous and more reproducible, how to apply for grants, taking these questions into account. So they are concerned. Now, a lot of people may think, and most headlines focused on psychology, but is this really about psychology? It turns out it's not. It's not specific to psychology. It's in many aspects, many fields of science. So here is an example on brain imaging, a paper that discusses issues with replicability. Behavioral genetics we already mentioned. This is actually the white paper, the summary paper resulting from that conference I mentioned that was held in Israel. But even in cancer research, there are issues with replicability. Economics, it's not about just biology. Even fields like economics, predictions and analyses and interpretation of data, all of these questions come up, and it turns out that it's not always replicable what people have been found or have been said. So here are some publications that discuss these questions in general about scientific problems in general in science about reproducibility or replicability. So I highly recommend you take a look at these. Clearly, it's not just psychology, and definitely not just zebrafish. So what the heck is going on? Are we scientists unable to do research? Have we forgotten how to do good science? So it's an interesting question, because if you look at how many publications are produced per year, what we see is that recently we are producing a much more larger number of publications. And these publications are a lot more easy to access to all of us due to electronic technologies. And so we are much more scrutinized also. Scientists look at other scientists' papers much more quickly. We often read these very fast. We can start doing control experiments, trying to figure out who did what. And so there's a lot more scrutiny over what we do. My feeling is that actually there is no crisis. We just have more studies and a larger number of mistakes as a result. Now, some people might argue that these mistakes are disproportionately larger, the number of them. Some say yes, but maybe we are just better connected and thus more transparent and better scrutinized. Now, that of course doesn't mean that we don't need to improve. The question is, what's wrong? What do we do incorrectly, and how we could correct the issues? So this is what the talk is going to be about, to kind of review general questions, and after that, a bit more specific to zebrafish about what may be creating the replicability, reproducibility issues, and what we could do to improve. So first, I will talk about the general issues, kind of broadly speaking, and then I will go to the unique challenges specific to zebrafish. So this is my own personal list. Some other publications organize things differently or emphasize different aspects of the replicability crisis. But from my perspective, these are four somewhat overlapping, not completely distinct problems that may lead to replicability, reproducibility issues. Number one is publication policy. So probably all of you experienced that there's a bias against non-significant results. There's also bias against not novel studies, that is studies that just aim to repeat someone else's or your own work. So we don't like to do that. And I will actually talk about these a bit more detailed manner. Statistical analysis related issues. That's another whole set of interesting questions. This is a particularly interesting one, the asymmetry in statistical inference. I won't have enough time to actually talk about this in detail, but I will just bring up a simple point about this. The other is there are many different ways you can do statistics. Almost all of us use the so-called frequentist approach, the classical statistical approach, but Bayesian statistical approaches are gaining foothold because they address many of the dichotomous issues, dichotomy, yes or no, significant or not type decisions that come with the frequentist approach. So I will not go into this, but I wanted you to be aware that depending on how you run your statistics, you can actually conclude very differently. And of course, that will influence how well you can replicate, reproduce your studies. Sample size, of course, very simple. Often we use too small sample sizes, and if we are lucky, we get something interesting, but is it reliable, is it not? That's a big question. Now, p-hacking is another interesting point. I will, if you don't know what it is, I will give you an example of that. That's a big problem also. I will also talk about inappropriate correction methods for type 1 or type 2 error. If you don't know what type 1 or type 2 error is, I will just briefly define it. It's a very basic statistical concept we all need to be aware of. And depending on how you correct for these errors, you may or may not be able to come to the right conclusion, and therefore you may influence the replicability of your results. Now, this is a very important problem, inappropriate control for known confounding factors, which also basically includes inappropriate randomization across different factors. Perhaps the most vexing issue is that, especially for zebrafish research, unknown confounding factors. That is, insufficient understanding of what may influence our results. This is a huge problem, and I will spend a lot of time on this explaining what I mean by this, what kind of factors we may not know about, and how we can actually take care of this issue. All right, so here is an example from my own personal scientific life, publication bias by referees and editors. What referees and editors tell us when we submit a publication for a journal. So here is one example. This is an email I received from referees, and the editor basically agreed with them more or less. But if you look at this, it's kind of hard to read on a small screen, but let me just read this area. Neither of these variables influence the behavior measured. So the referee is complaining that it's a negative result. And as a result, this study does not contribute to our understanding of developmental ethanol and FASD, fetal alcohol spectrum disorder. So the referee is complaining that we found negative results, and therefore this study is not interesting. It doesn't contribute to our understanding of ethanol effects and how ethanol affects developmental processes, and it's not relevant for this reason for FASD research. Now, the other referee doesn't say that. He actually, or she specifically states the findings are negative but interesting, allowing to exclude some of the potential causes of social deficits reported earlier. So the referee got it perfectly. This is actually what we try to emphasize, that yes, negative results are very important, and not just when you want to make sure that everyone knows that something didn't work, but in this case, it allowed us to interpret prior positive findings. And this interpretation is very crucial. So again, one referee is the old school. Negative results are not good. Other referee is actually on line with basic what we are saying. So often you encounter these problems when you try to publish your paper, and sometimes the editor actually just doesn't take the time and says, okay, well, it's negative results, not so interesting, so sorry, study is not accepted. Luckily, in this case, we were able to persuade the editor that yes, indeed, this is an important finding. So we basically published this research study in alcoholism, clinical and experimental research. And the title of the publication is Behavioral Responses to Novelty or to a Predator Stimulus are not altered in adult zebrafish by early embryonic alcohol exposure. What was the point about this? Is that we thought that maybe anxiety, maybe anti-predatory behavior, fear, anxiety got altered by our alcohol administration, and that is the reason why these fish shoal form groups differently. Turns out it's not. So it's a very important piece in the puzzle that allows us to interpret our social behavioral deficit findings a bit better. All right, so this is just me trying to be funny. But it's actually true. We also have a bias. Ourselves, our authors, we are biased against publishing old stuff. Just like animals, we are all interested in finding new things. So here is a T-maze spontaneous alternation task that I developed many, many, many years ago for rats and mice. And basically what it shows is that practically anything and any species and any circumstance, if you test animals, they will always prefer seeking novelty. They will always prefer looking at, smelling, checking out novel objects, novel areas. In this case, the novel area is the other side, this side of the maze. The animal goes to the left side, and then it's allowed a free choice, and usually it goes to the right side. We do the same. We want to find new things. We want to find new results. We don't want to just repeat old stuff. So there is a bias among us too. Now, this is really an illegal thing to do. Again, it's a type of publication bias. We call p-hacking. So p-hacking is when we publish only positive, meaning significant results, without mentioning non-significant results. So we focus on the interesting stuff. So just to warn Dennis, this is not actually real data, it's just an example. So imagine what you have here is two groups of animals. One is called group X, the one is called group Y. And then here you have, let's say, 6 behavioral measures, A, B, C, D, E, F, whatever they are. There are different ways to measure behavior. And this is the actual scale on which you measure behavior, the y-axis. And as you can see, there is some variation. Some behavior measures really don't show anything. But this one, behavior measure C, shows a nice group difference. So a clever scientist would say, well, okay, let's publish this because this is positive. This is interesting. This is significant. So that's a major problem. That is what we call p-hacking. When you discard non-interest, so-called non-interesting, that is negative, non-significant findings, and only focus on the one that you think would be most interesting. So again, there are two ways of doing that. Submitting only studies that found significance and burying those that didn't, the major publication problem, because a lot of people test, let's say, drugs, mutation effects, all kinds of things, all kinds of manipulations, and they find negative results, and then they don't publish it. So how do you know about those negative? You don't. The problem is you don't know about it. You want to run a study checking out the drug effect. Let's study the effect of that drug in zebrafish. And then you do the drug study and you find it negative. And a previous publication of such negative results should have saved you, would have been able to save you from the trouble and publishing it or repeating that negative experiment. So it is important to publish negative results for that reason. Now, the other problem, as I mentioned, reporting on dependent variables, only dependent variables that showed significance, and ignoring those that didn't show significance. That's also a major problem because really it doesn't allow appropriate interpretation of your results. And if you measure, 100 variables, out of the 100, if you keep the p threshold, the probability of null hypothesis being rejected 5% or being correct 5%, then out of the 100 variables, you will find 5 showing significant effects. And then if you just present those, then of course you are not correct. So you cannot do that. You have to basically present everything as you found. You are not supposed to pick and choose based on significance. So avoid p-hacking, that's the point. Now, when I mentioned this so-called crisis that I don't think actually exists, but still there are problems, a lot of people started to talk about this, and things started to change for the better, in the sense that the more editors, the more referees, the more authors are aware of these problems, the better we focus, the better we take care of these things. So here's an example of a study in which they explain promoting transparency is important, meaning make your data available, publish results as they were found. Don't pick and choose. Publishing replications, registered reports. Now that's an interesting one because basically what it means is that you pre-register your methods, meaning what behavioral measures, in case of behavioral analysis, you are going to measure, what are your methods, and what you are expecting to find. So you register these in advance, and therefore, whatever you find, people will know that these are the methods that you followed, and you didn't just pick and choose after the fact, after you have already found the results. So this actually guarantees that you basically avoid p-hacking. And of course, the paper mentions that null results are supposed to be published, that is negative results, when you don't find significance. So this paper was in behavior neuroscience, so it's specifically good for our particular topic, behavior neuroscience, psychopharmacology, zebrafish behavior research. All right, here's another paper in +1, why publishing everything is more effective than selective publishing of statistically significant results. I already mentioned it. It saves a lot of wasting of effort if you have to keep searching for what was negative, what was positive, because no one publishes the negative results, and you have to redo those studies. That's a huge waste of your time and money. Here's another example of an effort to kind of increase replicability and reproducibility. The Bill and Melinda Gates Foundation. Gates Open Research, so a platform for rapid author-led publication and open peer review of research funded by the foundation. So open peer review and open publication means that you don't wait for the referees to respond. You publish your study as you wrote it, as best as you can. And then referees, people who read the paper, comment on it. And they publish the comments with you. And it's all open, and you can view it in real time, basically how things are developing, how people are commenting, what kind of positive and negative comments they make. So this, again, basically promotes transparency of science. I want to draw your attention to this particular journal, F1000, which is a very interesting model, exactly following this type of openness, open publication format. All right, so this is a somewhat complicated issue, statistical inference. So I wanted to just give you a very brief example on how do we interpret p-value of, say, 0.09, which clearly is not significant. It's not smaller than 0.05. It means lack of significance. But what does it really mean? Some people, when they find this, state, actually, you can find publications, many publications, that this means that this is proof the treatment had no effect, or the groups of animals did not differ from each other. But this is not actually what it means. A lot of people interpret this way. Is this really a correct interpretation? So look at the probabilities. 0.09 means that the probability of the treatment having an effect, or the probability of the groups of animals differing, is 91%. So it's pretty high. Okay? So it's not a proof for the treatment having no effect. It just means that you could not find a significant difference. It doesn't mean that you proved a lack of effect. So let me just go to the next slide, which makes the point a bit further and perhaps more clearly. There is this asymmetry in statistical inference. That our statistics, that is the frequentist approach, is designed to test presence of an effect or difference with high certainty. Meaning, if the presence of the effect or difference is 95% or more correct, then we accept it. But it's not designed to prove the absence of the difference or the absence of the effect. So in other ways, to say it in another way, a null finding doesn't mean that you have identified the lack of effect. So you can word it this way: a significant absence of effect would require an equally stringent P threshold, that is, P being at least 95%. The point is, this is where we accept, hopefully you can see this on your screen, this is where if your P threshold is this, 0.05, if your phenotypic value is here, that is closer to the right end of this distribution, probability distribution, then the chance of you of the null hypothesis being correct is very small. So then you accept this as significant. You could apply the same principle if you wanted to prove lack of effect, but then you would have to be here within these two lines, basically. So that's the 95% probability value. At least 95% you should be, and that would be the proof of lack of effect. Now, what has it got to do with replicability? Remember, the issue with replicability is that we are not talking about all we care about is finding significant results. So we have this gray zone between here and here where things are really uncertain. You're not proving the lack of effect, and you're not proving the presence of the effect. So this is a large chunk of uncertainty that comes from basically the asymmetry of our statistical inference. And therefore, when we try to repeat things, sometimes we end up on this side of the probability distribution, and sometimes on this side of the probability distribution. So a bit complicated, but again, the point, this is all about the asymmetry of the statistical inference in frequentist approaches. Bayesian approaches take care of this. Take care of this very differently. So I highly recommend that you explore those statistical procedures and employ them in your future research. All right. So this is a bit easier to explain, simpler problem, but still we make this mistake so many times, and I have seen so many papers in which we see these problems to occur. So type 1 error is when one thinks the results are significant, even though they are actually not. Type 1 error in statistics means that when we think the results are non-significant, even though they actually are significant. So there are reasons why we commit type 1 error. That's when we have many tests or many measures that we employ, and therefore the experiment-wide error rate is the p-value times the number of measures or number of tests we employ. So we inflate the probability, and we think we didn't. So we find a result significant when in fact it wasn't. The other side of the coin is when you try to correct for this type of error, but you use a too conservative, too stringent correction method. A lot of people employ Bonferroni. Bonferroni correction is a brutal correction. It basically makes it very difficult to find a significant effect, and it actually creates the opposite problem to the type 1 error. It creates type 2 error. That is, you will find results non-significant even though they are actually are significant. So what's the solution? Many of you probably know that the best way to take care of that is to use a post hoc, a type of post hoc test that deals with multiple comparisons differently. One of these is Tukey honestly significant different or range test. So this is a balanced correction method that doesn't inflate type 2 error and takes care of type 1 errors. It's a nice compromise. Another one, which a lot of people perhaps don't know, is this, the Holm Bonferroni method. It's a very simple correction method. It's a variation of the Bonferroni method. It's a manual correction. You can do it, a very simple calculation. Look it up if you don't know what it is. I highly recommend everyone use it because this allows you to balance type 1 and type 2 error, meaning you will reduce type 1 error without inflating type 2 error. So I highly recommend you pay attention to this Holm Bonferroni correction method. All right, so the next slide brings up an interesting problem that is a specific example of finding not replicable results. Crabbe and colleagues, Bruce Dudek and Douglas Walston and John Crabbe, published a fairly famous paper by now. It became basically like a milestone, very famous paper in 1999, where what they did was they coordinated all their experiments. They had 3 different sites. So Crabbe is in Oregon. Bruce Dudek was, I believe, in somewhere in the middle of the U.S. That time, and Douglas Walston in Canada. Completely different laboratories, different geographic locations, but the investigators coordinated everything. They made sure that every procedure, every aspect of their experiments were done in an identical manner. Even the timing of the experiment, because they were roughly around the same time zone, they timed everything perfectly to the minute, basically. They used the same equipment, same apparati, same procedures, and they made sure they used the same genotypes of mice. So here are some examples. Here's an open field they were using. Here's a plus maze. A couple of arms are open. There's a classical plus maze, and a couple of arms are closed. They used the rotating rod. They used the water maze, and they used ethanol drinking self-administration paradigm. All organized, made exactly the identical across the 3 laboratories. And what they found was really surprising. They found a significant genotype environment interaction. So what did it mean? Genotype environment interaction means that in one laboratory they found some differences between genotypes, and in another laboratory they found this difference either non-existent or different. So depending on in what laboratory they measured the different genotypes of mice, they got different results. So they basically got lack of replicability. Actually, it's reproducibility, not replicability. So let me just correct this. So they found lack of reproducibility, they really did a good job discussing why their results did not replicate. But what turned out to be the case is that they slightly missed an interesting pattern in the data. They did bring up this point, although in a subsequent publication, but there was the interesting pattern in the data. Tests, behavioral tests that required no or minimal human handling appeared to be quite reliable, meaning these tests provided replicable results. But those tests that required a lot of human handling did not. So I was listening to prior presentations, and for example, Sue Guo brought up an interesting point. They try to avoid human handling, and there's a good reason for it. It's very difficult to standardize human handling. So let me make a point. What happened in the Crabbe study, when they went back and they tried to analyze, tried to figure out what could it have been, why their results were not replicable. What they found was that everything they did exactly the same way, but there were certain things they couldn't standardize. So in one of their laboratories, the experimenter was allergic to rats and mice. So this gentleman wore literally a hazmat suit, a complete olfactory isolation from the mice, because he didn't want to get allergic reactions. But that means also that he was isolated from the mice, but the mice were also isolated from him from an olfactory standpoint. The mice couldn't smell him. In the other laboratory, the female technician was very friendly with the animals, was an animal lover, had mice at home, and handled the animals in a very professional, very nice and gentle way. So there were differences, big differences in how handling actually influenced these animals. And although everything was standardized, this aspect of the behavioral procedure was very difficult to standardize. And the experimenters kind of missed that. And the result was lack of replicability of many of their tests in which human handling, anxiety, fear could be a driving force. So, all right. So I wanted to bring these up as a general problem because it's not just, it's really not specific to zebrafish. A lot of people, a lot of scientific fields struggle with procedural methodological issues like these. And so it's not that we zebrafish scientists have more problems, but we do have some very important problems to take care of, primarily because zebrafish is quite new in our field. So in behavioral neuroscience, behavioral genetics, psychopharmacology, zebrafish is relatively new. And so as a result, our job is to actually improve and figure out all these problems that might have already been figured out for the mouse or for the rat. So we have a lot of work ahead. And I'm going to mention a few examples. It's not going to be a comprehensive review of the literature, but a few important examples which I think we need to focus on. So one thing I already mentioned, handling-induced fear and anxiety. This is a major, major problem in zebrafish research. Some people may underappreciate it. Those who work with zebrafish, especially behavioral scientists, we know that this is a major problem because if you don't handle the animal, if you chase the animal with the net, if you expose the animal to open air too long, if you don't minimize these stressors, your entire experiment will be basically dependent on fear and stress responses and not what you want to do. So another important area where we need to improve is zebrafish maintenance and breeding. I will spend a bit more time on this. Despite the decades-long experience with zebrafish, surprisingly little is known about really what is the optimal way to maintain the fish. Another important point is genetic makeup. Genetic makeup, the genetic background of the animal can be a major confound, and sometimes we don't even know about it. So this is an important question. Experimental procedures, of course, how we do our studies. I will bring up a few kind of silly, sometimes not so silly, personal examples of how things can go wrong, what we did inappropriately, how trivial problems we sometimes created to ourselves. And the last point is, this is a general point, the pros and cons of standardization. Should we or should not we standardize? What are the advantages of one versus the other, and what are the disadvantages? Okay, so hopefully you guys can hear me, and everything is good. So I will go on. Let me just check if everything is okay.
Yes, we can hear you. Yeah.
Good, everything is good. Thank
Yes.
you very much for
No
confirming. Wonderful. All right. So let's see. Fear and anxiety as a major confound in zebrafish behavior research. We've been keeping zebrafish for a long time in the lab, decades, 4 decades. So some of these standard strains that everyone uses have been bred in captivity for multiple decades. Yet, I don't think zebrafish is domesticated. Not in a sense that rats, particular rats, but even mice are. The reason why I'm saying that is that there has been no unintentional or intentional selection for tameness. Meaning, the zebrafish will regard you, the experimenter, as a major predator when you are trying to catch it and trying to chase it around and fish it out from the home tank and put it into the testing. So this is a big problem. So human handling remains a big problem. It means chasing, catching the fish, exposing the fish to hypoxia, open air, and also exposing the fish to abrupt water parameter changes. If your water parameters differ between the home tank and your test tank, that's a major problem. So what are the solutions? Well, Sugu already mentioned one. Don't handle the fish. Try to design experiments where you do all your tests in the home tank. Now, if you cannot do that, if you have to handle the fish, try to handle it with utmost care. Minimize chasing. Now, this is very difficult to define because it's almost like, it's impossible to describe what it means, but briefly, it takes a lot of practice. You have to be very slow, very gentle when you move the net. If the fish jumps, escapes from you, you have to wait until it calms down. So students who have fish at home or who have been working with zebrafish in the lab for a longer time, they develop this ability. So it's very important to practice. Very important to figure out how the zebrafish move, what it means when the zebrafish is trying to escape from you, what kind of behaviors it exhibits. So you need to read the response of the zebrafish and react appropriately. So you have to handle them with care, you have to minimize chasing and stressful catching the fish. Minimize out of water time. So if you carry the fish from one location to the next, don't just net the fish and carry the fish in the net. Put the fish into a beaker, move the fish with the beaker, and then put the fish from the beaker basically into the test tank. So that's one way to kind of minimize out of water time. And of course, you need to control and equalize home tank and test tank water parameters if at all possible. So that's very important. Now, if you study, for example, learning and memory, and you worry about, like us, we worry about handling related issues, we always implement elaborate habituation protocols. The problem with them is that if you don't do them correctly, they lead to the opposite effect. Instead of the animal habituating to the procedure and the new tank, they actually get sensitized and they become increasingly afraid, increasingly anxious, afraid of the procedure and the test situation. So it's a touch and go. It's a very difficult thing to do. Sometimes we are successful with habituation. Sometimes, unfortunately, it doesn't work. So it is, again, an open question how to do that, but it's one that you have to focus on, and eventually we have to find a nice way of solving it. Again, one obvious solution to all of this is home tank tests. I have to emphasize this is really an important way to go. I think it will be increasingly utilized in zebrafish behavioral studies. It's very important to develop tests that you can conduct in the natural home tank where the animal lives or grew up. It will solve all of these or most of these human handling related issues. All right. So the next topic. This is about maintenance and breeding, and most of us believe that we know how to breed fish. We know how to maintain the zebrafish. Well, it's not exactly right. Actually, it's not true. So a lot of people notice that we don't really know how to do it, and even if we think we do it differently. So there's a huge variability from one laboratory to the next. So here is an example of a meeting that was organized in 2017 by the NIH, the Office of Research Infrastructure Programs. And they invited a lot of zebrafish scientists. They call them the leaders of the scientific field or influencers of the scientific field. And basically, a bunch of us got together and then discussed all kinds of questions about how to maintain fish. What are the standard parameters within a range of parameters, ideal parameter range within which we should keep our fish? Why are there differences? What are their differences? What differences there are among laboratories, and should we or could we minimize these differences? So the point is that it turns out that most of our methods, the ways in which we keep fish, are the results of trial and error and not systematically controlled studies. So very few studies have been conducted that would investigate the effect of water source, salinity, salt composition, pH, temperature, light level and wavelength, tank size and fish density, food type and feeding frequency, whether we use enrichment, or what kind of pathogens are in the water, and how we breed the fish. Many of these questions, most of these questions have not been systematically analyzed. And so, depending on what your preference is, one facility performs this way, breeds this way, keeps fish that way. Others do it differently. So there's a huge variation across facilities, and there's very little consensus how to do this. All right. So just examples: water source and treatment. Some people use municipal water from the faucet. Some people use water from a river. Many of us use reverse osmosis filtered water. pH. Some facilities have 6, others 9 pH. Salinity, again, a huge range. In some facilities, you have 100 microsiemens water in which the fish are living, and in others, 3,000 microsiemens. So it's basically almost like ocean water. So huge differences. Salt composition. Most of us use sea salt, but the problem with that is it's sodium chloride, mainly sodium chloride, and it's very unnatural for zebrafish. So some people argue that maybe we should use calcium carbonate, magnesium carbonate, that are more natural in the habitat of, that are actually the salts that are found in natural waters of zebrafish. So again, the point is that some people use these. We started to actually use more calcium carbonate-based salts in our zebrafish maintenance, and less sodium chloride-based salts. But again, there are huge variations. So this is a big problem, and this also makes results difficult to replicate, because if you keep fish in one lab this way and keep fish in the other lab differently, these maintenance condition differences can induce big changes, big differences in behavior and brain function that you study in your fish. All right, housing density, another important question. Enrichment and tank size. Some people, most of us actually, you keep zebrafish like this in the standard high density zebrafish rack. a lot of zebrafish in a small tank. This is how they live actually in nature. They form shoals, yes, they're close to each other, but not as tightly, not as many crammed into a tiny space. They have a lot more space. They scatter around, they move around, a lot more space. They have with plants, a lot of enrichment in the natural habitat, but not in the lab. So these are important questions that we need to resolve. What is really the optimal way to keep the fish? And should all of us keep them in that optimal way? All right, genetic makeup. So it's a very important problem. Sometimes the origin of the fish, the genetic makeup of the fish, the strain origin, is not even reported. Now, there's another important problem. I will explain what this is. Genetic markers versus strains. Sometimes these terms are mixed, and the result is devastating. Another big problem with our zebrafish studies is that we really don't have inbred strains. We have quasi-inbred strains. And so we can suffer from what we call genetic drift. I will explain that a bit later. The other is sometimes there are mistakes. And I will give you an example. And this is the question about that. Is AB always an AB? And then you have the question of whether we should use outbred versus inbred strains. All right, so I will make this simple, but basically what happened in this study is that these investigators, and I usually don't do this, but I hate to call them out on this, but this is a major mistake that these particle scientists made, and I hope no one else will repeat that. They claim that they found strain-dependent ethanol effects. They think that they are studying 3 strains. What they are studying is not strains. What they got was zebrafish from pet stores, and one group of fish had the wild-type stripe pattern. The other group of fish had the leopard phenotype, the dotted leopard phenotype, and the third one, well, they were blue longfin. The problem is that these markers, the stripe pattern, the leopard, and the blue longfin, represent only a few genes. They do not define the genome of this fish. You can have a leopard mutant on an AB background, or you can have a leopard mutation on a Tübingen background, or you can have a leopard mutation in an outbred background. Same way, blue longfin is probably 3-4 loci, and so whatever the genotype of these fish are is completely unknown. And it's not even that. Likely, if you buy a leopard fish from a pet store one time, and then you go to another pet store and buy a leopard fish from another pet store another time, the genetic background of these fish, the leopard fish that you purchased at 2 different places, 2 different time points in your life, could be entirely different. To think that these phenotypical markers define strains is really a gigantic mistake. And again, the problem is that, yes, there is a leopard inbred strain, and there are wild-type pattern strains that are genetically fairly homogeneous, but then these are strains that are obtained from scientific facilities, whereas these gentlemen, ladies and gentlemen, got their fish from pet stores, completely genetically undefined, genetically random, variable populations of fish. So it's a major problem. Mixing up these terms is not just semantics, not just how we use the terms strain versus not strain, but interpreting the results like they did, that they found genetic strain-dependent alcohol effects is completely wrong. The other problem is how inbred our strains are. Most standard zebrafish strains, A, B, T, TU, WIC, are not inbred at all. So they are about 70 to 80% homozygous, which means that they have a huge number of loci in a heterozygous form, which means that if you breed fish like this, you will experience genetic drift, unless you have an infinite number of fish in your facility, and you don't breed them multiple times. So the point is that random genetic drift creates laboratory-specific genetic strains. So what you call standard strain may differ from one lab to another. It's a major problem, potentially major problem. What I always recommend we do is to disclose how many generations we bred the particle AB. Where was the source? Where did these ABs came from? Where did our standard strains come from? It's very important to specify that because that will allow others who read our papers to see how potentially different our AB or other strains may be from theirs. Okay, this is just an example for an interesting point that a lot of zebrafish experts say that it's impossible to inbreed zebrafish. It's not possible to make fully inbred zebrafish strains. It's not true. Here is a publication where they didn't use anything fancy, didn't use gynogenesis, didn't use any specific genetic techniques. They simply brother-sister mated zebrafish. It's called SIB mating or SIB pair mating. And so basically they bred zebrafish together, brother sisters for a long time. After 15, 20 generations, you get 99% homozygosity. That's what they basically created. They created two zebrafish strains that are almost fully homozygous. It is possible. I recommend we pay attention to this. And instead of just accepting the fact that we don't have inbred strains, we will start breeding inbred strains. We should be starting breeding inbred strains and creating a genetically homogeneous strain set that allows us to maintain these fish without any issues with genetic drift and so on. All right, so I will zoom through this a little bit fast because we are running out of time. Unfortunately, we ended up with a problem. We got ABs from another source. It turned out that there was a genetic contamination. We learned about this after the fact when we started to breed these fish. We realized that these are not pure ABs. So it's very important to be absolutely careful not to mix, not to interbreed strains, and always check whether your particle strain is indeed has that genetic makeup. Using genetic markers, you can double check whether your ABs or Wicks or Tübingen TUs are actually indeed the right strain. Okay, this is an important point. I wanted to spend a little time on this. So most people assume inbred is better. I just explained why inbred strains are better. Yes, they are from one perspective. They give you that genetic stability. Stability across generations, lack of genetic variance leading to smaller phenotypic variance. But this is actually not such an obvious question. There are some advantages of using outbred, genetically variable populations. One important point is that these fish, the outbred fish, you could consider species typical. That is, not like inbred strains that are unique. They have unique, genetically unique features that manifest in the phenotype. So they are not strain idiosyncratic. These outbred fish are fairly typical of the species. And then another interesting point, and I won't have the time to really explain this, but often outbred strains show reduced phenotypical variance. And this is due to better buffering against environmental variation, also known as genotype-environment interaction. So outbred populations can actually be quite useful. All right, I will actually skip to the personal stories. These are interesting, but we won't have the time. We made many mistakes. One of them was not ours, it was someone else's. We tried to replicate or use nicotine doses that others used, and it turned out that actually this dose, and even fraction of this dose that you see here, even 50 milligram per liter bath concentration killed our fish. So we ended up actually having to use 4 to 8 milligram per liter, much lower. We suspect that what happened with the study here is these two studies is that the nicotine that they used, nicotine ditartrate, was old, was decomposing basically. So it wasn't efficacious enough. But of course, we learned about this only when we tried to replicate the results, and our doses turned out to be orders of magnitude smaller than theirs we could use. So I'll skip through this. This is an interesting one, very trivial, very simple. These are the images to which zebrafish respond really well. And accidentally, a student presented images like this, and of course, zebrafish didn't respond the same. Another important point, I bring it up now because in a previous presentation, we heard about this, and I know actually multiple presentations, that we use something called light-dark task. But we often mix terms. When we say light-dark, we sometimes mean well-illuminated versus not illuminated. And sometimes we mean white versus black background. And sometimes we mean closed versus open. I emphasize these are three distinct factors. So if you're interested in a study that started to explore these different factors separately, here is one. There are actually more than this, like 4 or 5 publications on this topic already. That basically tells you that we cannot just simply call it light-dark. It's the level of illumination that is different, that drives behavior differently, and the background shade drives behavior differently. And whether the tank is closed or open makes also a difference. So depending on the combination of these factors that define your task, you get very different preference responses. Sometimes you get dark preference, sometimes you get light preference. So pay attention to this. These are not the same factors. These are 3 independent factors that you need to pay attention. Okay. All right. This is the last point I want to talk about, and that will conclude my presentation. Should we standardize? This is a trivial question. It seems trivial. Most assume that the answer to this is, of course, we should standardize. Standardized maintenance conditions, standard genotypes, standard procedures should all aid reproducibility within and across laboratories. Standardization allows better control of the environmental factors and genotypes, better understanding of what may influence our results. In general, should all allow us to reduce error variation and thus increase precision and statistical power. That's all good. This is really important for reproducibility. If you standardize, if you do everything the same way, you should be able to reproduce results. Using the same methods, the results should be identical. But how about replicability? Remember what we defined at the beginning? Reproducibility is obtaining the same results repeatedly when using identical methods. Replicability, on the other hand, is reaching similar conclusions even when variations in methodology. So standardization will definitely aid reproducibility, but replicability is another question. Take a look at this. There are a huge number of publications discussing this problem. Why do most clinical trials fail? And there are many reasons for it. Here is one publication that lists up many of these, but in reality, many publications miss an important point. Standardization leads to reduced error variation and thus increases statistical power. That's great. Increased statistical power allows finding efficacy in preclinical studies. That's also great. So when you are studying, let's say, the effects of drugs, the effects of mutations in an animal model of a particular disease, for example, you find significant results. Fantastic. The problem with that is that real life is different. Real life human populations are not genetically homogeneous and do not live in controlled laboratory environments where the environmental variation is reduced. In other words, standardization is good for reproducibility, but it's not good for replicability. Okay? So hopefully you guys will think about this and understand what I mean. Again, the main point, if you standardize everything and everything is done exactly the same way, you are creating an artificial situation, which basically is very different from real life. And if someone modifies the methods a slightly bit, you will get different results. So some people actually argue that allowing variation in results, variation in methods, and then looking for still common findings is the way to find the most robust, most replicable result. All right, so for basic research, the discovery process should start with standardization. It is very useful. Understanding the factors that may affect our results and being able to control them or randomize across them is very important. So this allows us to establish fundamental discoveries that are reproducible. But allowing variations, reducing the requirement of standardization, may allow discovering features, phenomena, processes that are robust against the effects of such variation. Thus, for basic research, basic discovery research, blue sky research, standardization is highly recommended. But this research may be followed, should be followed by loosening the criteria of standardization and allowing the discovery of more broadly applicable phenomena useful for translational or clinical research. Okay, so this concludes my presentation, and sorry I'm going a bit over time. I wanted to thank you, Dennis, for inviting me. I wanted to thank Barbara, Vanessa, and everyone else involved for organizing the meeting. I really appreciate the speakers for participating and making their effort during this difficult time. I would like to thank you all participants for listening to us and asking good questions and making the meeting a success. And I would like to ask you all to stay safe and be optimistic. This will be over, not just my talk, but the pandemic as well. So hopefully we will meet live and see each other and talk live again very soon. Take care, and I'm open to questions now.